All right, so part 3, sampling mobile organisms, a little bit backwards. We're going to talk about some general methods for sampling mobile organisms and then invertebrates and aquatic insects in particular. So the first general idea is you always want to standardize your methods. You want to make sure that you're doing the same thing across all of your collection methods. You want to assess the condition of each organism that you capture. That can be really important data. Are they damaged? Have they been injured? Are they reproductively active? These are all really important measurements. Do they have infections? How big are they? Their size and their weight. You might want to take a DNA sample, blood, or a small piece of tissue or hair. You might want to take some photographs. Depending on the type of organism, if it's an amphibian or a reptile, you might want to measure its snout to vent length. So from the tip of its nose to basically the tip of, not the tip of its tail, but where the tail meets the body, snout to vent. And you can also do a lot of behavioral work with animals, with mobile organisms. It's a little bit harder to study plant behavior, although there are a few behaviors that plants utilize, but animals have more interesting behaviors. So there are 4 different approaches to studying behavior. One is causation. So what factor influenced the onset of the behavior? Like what causes them to act that way? That might be one question you might ask. Development. How does an individual develop the behavior? When does the behavior start? Is it something that an adult does or a juvenile? Function. What is the behavior for? Does it serve a purpose? And evolution. How did the behavior evolve? Did it, animals used to do something differently and they've evolved new behaviors? These are some really cool questions you can ask about organisms. You can set up what are called choice tests or buffets for organisms to see whether they choose a particular item or not. You might be interested in studying family structures. How do the families work? How do extended family members interact? Dominance hierarchies, who's the dominant organism? Is it a dominant male? Is it a dominant female? Are there, males or females that are involved in the family structure but not dominant? So lots of interesting things you can ask. What kinds of interactions are happening with organisms of their own species and with organisms of other species? And then, are they acting aggressive? Or are they acting curious? What kinds of behaviors are they exhibiting? Okay, so moving on to invertebrates. Invertebrates are organisms without a backbone. And so what's kind of nice about working with invertebrates is that you don't generally need additional permits to work with invertebrates other than like the permit to access the site or to do research at a particular location. Invertebrates don't generally require additional handling permits, except when they're endangered. And so that's one nice thing about working with invertebrates. They are also an exceptionally diverse and abundant group of organisms. So they're everywhere. They tend to be found in large numbers. They're relatively easy to study compared to, like, say, large predators, which you might wait your whole life to see one, right? So you can make observations of large organisms, things like, grasshoppers or butterflies. And for example, this is from the book, you might use a W-shaped transect walk to make counts of butterflies when they're flying, say in the prairies around here. We have great butterflies. Might be an option to actually make observations about them and their behaviors. But smaller invertebrates are more difficult to observe and usually require that you capture them and sometimes require that you kill them to really identify them and study them. So they need to be trapped, collected, and then sometimes killed and preserved. So here's some examples for capturing aquatic invertebrates. So you can just go out and pick up rocks and look for them and then pull them off the rocks and capture them that way. You can use a variety of flat-bottomed nets. This is kind of like a D-net. Here's an aquarium net, maybe for catching larger bodied organisms like tadpoles or crayfish. Here's a really small net that you might use for surface film fauna. You would put these two nets maybe along the bottom of the stream and then either dig into the sediment or disturb the sediment above the net to get things to kind of flow into the net. And this is a SURBER sampler. You can see there's a frame in the front, and she's placed this in the stream with the water flowing in this direction. And then she would get in there and disturb this sediment inside the frame, and that would kick up all the organisms into the net that were living in the benthos, or the bottom of the stream. Here's a closer up view of the SURBER sampler with the water flowing into it and the collecting vessel at the back, and this nice kind of quadrat in the front that helps you to sample quantitatively from a known area. This is a similar sampling device called a HESS sampler. It has a round base, but what's nice about it is you can really dig it down into the sediment like a bucket. You can kind of dig it down into the sediment, and then again, you would want to swirl things around in the bottom and get stuff to get moved up into the net here and out into the collection device. Similar to the SURBER sampler is something called a drift net. Here's a big drift net that's been anchored with rebar into the stream, and then the net flows this way. The water's flowing into the net. This is not a net that you would go and disturb the benthos, but instead you'd be interested in catching everything that's just floating in the water column. So that's why it's called drift. So aquatic insects will occasionally just drift to move downstream, where they might get scared from their site and kind of float downstream. You might be interested in catching that community of drifting organisms, or maybe capturing organic matter like leaf litter as it floats downstream. Here's a series of drift nets that have been set up to sample across the entire stream. And then this is a picture of a standard D net. It's a little bit bigger than the nets I showed you before. The handle breaks down. And again, you would place this in the stream and disturb the benthos ahead of the net to get things to fall into the net. If you're interested in sampling lakes or large rivers or ponds, you might be interested in a plankton net. It's a small net with a very fine mesh here. It looks kind of like a kite. It has a collection bottle at the back, and you would throw it out and then pull it across the surface or just under the surface of the water, depending on what kinds of organisms you're sampling. From boats, you can lower plankton nets into the water and then pull them up through the water column to sample the zooplankton and phytoplankton that are living in the water column. If you're interested in sampling the bottom of a soft, like a silty or sandy lake, river, or pond, you might use a dredge or a grab. And so this is called an Ekman dredge, and this is a grab. They both have these kind of mouths that close on the sediment. They just have slightly different functionality. And so here's kind of showing an Ekman with the mouthpieces held open on these pins. And then you send a messenger down onto this this little device here that knocks the pins down, and these mouth parts snap closed. And you can see now this is snapped closed. And then you would pull it up, hoping to capture all of your sediment in the mouth of the dredge. These are called HESTER-DENDY samplers, and they're basically little bits of tile or wood that are put onto pins with different size washers to create different kind of habitats and environments for little organisms to crawl in. So you would place these out in the stream and you'd let them get colonized by, you could do algae or aquatic insects. And then after time, you would pull them out and you would determine who all has colonized these surfaces. Another option is to create your own material, like say you're interested in who colonizes leaf litter. You could make leaf litter bags. Here we are interested in leaf litter decomposition next to salmon carcass decomposition. It was kind of a smelly project, but kind of fun. You can see lots of different leaf litter bags going into the stream here in Utah, lots of different litter bags going into streams at Mount St. Helens. And so the methods basically have you collect leaves from organisms, plants, trees. Depending on, you know, what kind of leaf material you're interested in capturing, you might capture the leaves slightly differently. And usually take them back to the lab and dry the leaves, then weigh them and put them into these numbered leaf litter bags. These ones are made out of window screen mesh. Then you transport them to the field and you place them in the stream. And then over time, you might come out and harvest, you know, a fraction of these, like say 25%, and then you harvest another 25%. And then you have 4 harvest dates where you've collected them through time. And then you can bring the leaf litter back to the lab, dry it, weigh it, and burn part of it up to determine how much material was lost throughout the entire study. Here's just some close-ups showing leaves and tags in bags of different sizes. Sometimes you might be interested in taking samples for microbial analysis after the leaves have been out in the stream for a while and been colonized by fungi and bacteria. And here's a picture, kind of a blurry picture, showing a piece of rebar with a bunch of litter bags. And a method that I like to use is using color-coded cable ties to tell me when I'm going to go harvest which bag. So I might come back to this rebar and harvest all the red bags. And then the next time I might come back and harvest all the yellow bags, and then all the green bags, and then all the blue bags. So it's a way to keep from disturbing the other bags while you harvest the ones you're interested in. Here's a picture of a funnel trap close up. That might be used for measuring aquatic insects. I showed you pictures of these when we were talking about amphibians and reptiles as well, but you can see they can come into these little holes in the funnel, and then it's hard for them to get back out this little hole, and they get kind of caught in the corners. And then one last thing that I thought was really cool from the book is it talks about different ways to mark invertebrates. And so, you know, like you could imagine putting like maybe 10 spots of paint on your invertebrate, right? But that's only going to get you 10 organisms. But what if you want to mark and sample hundreds of organisms? Well, if you use this method, you have, this is kind of it's called the notum, the pronotum, mesonotum, and metanotum of an organism. Is kind of like the thoracic segment, and then these would be the wing pads. You can use the marks on the corners of the 4 corners of the thoracic area to denote hundreds in your sample number. And then you can denote tens on the left-hand side, elytra, kind of wing pad, and units on the right-hand side, elytra. So for example, this organism would be number 629. Why? Because it has a 2 and a 4 in the hundreds. So that gives us 600. It has a 2 for the tens here, and then it has a 2 and a 7 for the 9. And so cool. You can mark out from 1 all the way to 1,000, or probably 999, I guess. You can mark out 999 different individuals. Another way to do that would be to use different colors of paint. So if you have yellow equaling 1, green equaling 2, blue equaling 4, and red equaling 7, and you can have hundreds, tens, and units the same way. We could put a green dot and a blue dot to give us 2 plus 4 equals 6. Then a green dot here for the 20, and a green plus a red to give us 9, 629. So using this color method, you can also get 999 insects marked. So really pretty cool. And then you'd use mark and recapture methods to determine total population sizes. All right, that's the end.